Next topic is Golgi apparatus. So let's start with this one. In previous class we did the endoplasmic reticulum. Now we'll do the Golgi apparatus. So if you remember, I told you each and every organ, they are nearby the nucleus. Okay, like this ER here nearby this nucleus only, Golgi apparatus are also present. Okay, so what is the role of Golgi apparatus? See, main function is of it to modify the products, whatever the product is formed by the ER. Okay, whatever the product is formed by the ER, it is modified by the Golgi if any further modification required. It's sorted out, it do the shipping. Okay, shipping means what? See, suppose if we want to send something to a different address, what we are doing, what we are going to do, we just pack this item. Okay, whatever the item we are going to send someone on some another place, what we will do, we just pack that item. Okay, we put some address on it, then only it would be able to reach on that location, right? So here is also, if some products are formed by ER, first of all they are enveloped. Okay, they are enveloped. Okay, they are bound in a membrane, then they are sorted out. Okay, which to be sent inside the cell. Suppose this is the cell, so which this product, first of all it is bounded inside a membrane, then it is sorted out which where it is location. I mean, it should be sent inside the cell only or outside the cell to do its function. Then it is done the shipping. Okay, as per the its target. Okay, where need to be sent, then it's do the shipping and further secretions is done. Okay, that product is being secreted at that location wherever it is required, right? So this product is always formed by the ER. As I told you, ER is of two types. One is the RER, another is the SER. Okay, so RER is I mean, like ribosomes are attached on it, it make the protein, and SER make the lipids. Okay, so these two things, when they are getting formed, they enter inside the Golgi bodies. Okay, this Golgi apparatus, and further modification are done on these products, and then they are secreted in the form of vesicle, in this vesicle. Okay, and then whatever the location of it is, where are they required, they are sent over there, right? One more thing is what? Okay. Yeah, so Golgi consists of flattened membranous sac, okay, which is called cistern. So ye, I mean, like, just remember this thing, they are the made up of cisternae, like this one, sac like structure which are put or parallelly over each other. This is called cisternae. In Golgi there are actually two sides, one is the cis phase, another is the trans phase. So what does it mean? Let's have a look. So this is the Golgi apparatus, as you can see. Okay, always remember this is the nucleus. Okay, suppose I am saying this is the nucleus and it is always present nearby the nucleus, and here in from the nuclear membrane only this ER is getting formed like this. So just assume this is the ER. From ER the products are getting formed. Okay, once the products are getting formed, these products enter into this Golgi body for their further packaging. Okay, so from the surface from which it get enter, as you can see here also, you can see these products, they are getting enter inside the Golgi body. Okay, they are getting fused and the material is being inserted into the Golgi body. So the surface from which it is getting inserted, this surface is called the cis phase. Okay, okay, this is also called receiving phase, or it is also known as forming phase, from where the product get enter inside the Golgi apparatus. It is generally convex. It is generally convex in shape. If we'll see the shape of it, it is convex, like this, like this. Convex surface is this one. Okay, once it get enter inside this Golgi, so suppose this is the product, again if there is any further modification required, now let me know the modification. What are the modifications? Suppose RER forming the protein, okay, and SER forming the lipid, right? If you remember, in a membrane, if you remember, there are some sugar chain, if you remember, right? These sugar chain are getting attached to the protein as well as to the lipid, okay? If they are attached to the protein, then we call it as a glycoprotein. I have told you this one while when we did membrane. And if the sugar molecule are attached with the lipid, they are called glycolipid. Carbohydrate chain is attached with this protein, this is called glycoprotein. I have told you this one. And if it is attached with this lipid, then they are called glycolipid, right? Okay, so glycoprotein and glycolipid, they come together and ultimately make the plasma membrane, okay, because plasma membranes consist of lipid and protein only, right? So the attachment of the sugar to these protein and lipid respectively, this is called modification, okay? This is called modification. See what RER and SER, they are just making the protein and lipid. They are further converted into glycoprotein and glycolipid with the help of Golgi apparatus. What Golgi is doing? It is attaching some sugar chain onto these product and then make it complete. Then they are getting from glycoprotein. So this kind of modification is done by Golgi apparatus. Now did you get it? What kind of modification they are doing, right?
Yeah.
Sometimes sometimes very simple sugars are being created and they are further converted into complex sugar with the help of this Golgi apparatus as well. So such kind of modification are done on the products which are formed, right? So once the modification is done on this product, further they are just packaged, okay? A plasma membrane layer is surrounded by this, which surround this product, and they are just packaged. So this is what we called packaging, okay? Once they are packaged, they comes out from another surface. So can you see this one surface, this is called transphase. Okay, here it is written, this is called transphase. Yeah, another name is shipping side, or one more thing is also called maturing phase. So what is happening now? This vesicle is coming out from this trans surface and getting released in these small vesicles which are having these products. So you can see these are the products which are filled in this vesicle. Now this is secretive vesicle which are coming from the transphase. If their target is to do the function inside the cell, then they are getting inside only, and if it is target is to go outside the cell and do its particular function, then they go outside the cell. So that's why they are called transport vesicle, okay? So they go to their target wherever it is present. Yes, did you get it, beta?
Yeah, I got it.
Yes.
How they go to the target.
Yeah, so this is actual function of Golgi apparatus, okay? So now you must be knowing cis phase, which is also called as the forming phase, okay, and trans phase, which is called the maturing phase, or you can say the secretory side from where the vesicle secreted. Here vesicle secreted, okay, right? Now let's go further. So this is work done by the Golgi bodies. Always remember these two phases. These are very important phases, cis phase and trans phase. So many times question comes on this one. You must be asked about this one. What do you mean by this one? Okay. So what it actually do here, these are things written: modification in glycoproteins, manufacture macromolecule like pectin. Pectin is what? It is a polysaccharide. Okay. Do you know saccharides? Saccharides are basically sugar molecule, okay, like glucose we used to say. When a long chain of glucose is being attached in various form, one of the form is pectin, okay. When there are so many glucose are polymerized or attached with each other, then they are called pectin. Got it? It also involved in lysosome formation. See, the vesicle which is released outside from the Golgi body. If in this vesicle, the product which is filled in this vesicle, if the product is having hydrolytic enzyme, okay, if the product is having hydrolytic enzymes, right, then this is called lysosome. This is called lysosome. Okay. And if one more word is acrosome. What is acrosome? It is present in the sperm. Do you know sperm? That there are two terms, male and female gametes. So I mean, like sperm and ova. So in male, sperms are getting formed. This you will read later on in this syllabus. So in sperm there is a structure which is called acrosome. So this acrosome is also getting formed by the Golgi apparatus. Okay, so you just remember this one only, that acrosome is formed by the Golgi bodies. Okay, one more thing, we also called it as a Golgi body. Okay, we can write Golgi body as well, or we can say Golgi apparatus. Okay, got it? Okay, yeah. Let's go further. Next point is lysosome. As I told you just now, if the product is having hydrolytic enzyme, then this vesicle is known as lysosome. So it's a membranous sac of hydrolytic enzyme, that you have to remember. What it contains? It contains hydrolytic enzyme. What do you mean by hydrolytic enzyme? An enzyme which can digest what? Which can digest carbohydrate, which can digest protein, fat. Okay, so these all are things. So such an enzyme which is having the ability to digest all these kind of things, I mean, like it has a multiple enzyme. Like if it is I mean, digesting carbohydrate, it must have the carbohydrase. Carbohydrase is an hydrase in an enzyme. What is this? This is an enzyme which digests the carbohydrate. Protein, for protein digestion, protease enzyme is there. Okay, for lipid, I mean, like fat is also called lipid. Fat is also called lipid. So for this, lipase is there. Okay, so this hydrolytic enzyme is a combination of carbohydrates, protease and lipase as well. Okay, carbohydrate, protease and lipase as well. Okay, what are the function of it? This lysosome. First function is intracellular digestion. Okay, so see, whenever I mean, like cell is having food inside or something else inside, so their digestion is being done by the lysosome. Intracellular means what? Inside the cell. Inside the cell. I can write here also, inside the cell. Okay, this is called intracellular digestion. Got it? Like for example, amoeba, it do the phagocytosis. Okay, now what do you mean by phagocytosis? Phagocytosis, you can just I mean, like engulf and digest, or you can say just kill it. Okay, so what amoeba actually do? Suppose this is the amoeba, right? Amoeba is a irregular shape, it is not having a fixed shape, right? Suppose there is a food present outside this one. Okay, so what amoeba do? Amoeba will make a false feet. What we used to say, these are false feet. Okay, this is the nucleus, what I am drawing in a black dot. Okay, so this food is being taken inside the amoeba like this. Ultimately, what this food is being taken inside the form of a vesicle. Okay, now lysosome come over here. Suppose this is the lysosome. Okay, suppose this is the lysosome will fuse with this one. Okay, and release its whole hydrolytic enzyme inside this food particle and digest. So I'm just drawing a large view of this one. So what will happen? This is the vesicle, inside this the food particle, and lysosome also getting fused and releases its all hydrolytic enzyme. Now what it do? This hydrolytic enzyme attack over these food particle and digest it, and this is how amoeba do the digestion. It releases the nutrients and it absorb the nutrients. Okay, got it, beta? So that's how it is taking its food. So it make the food vacuole. Here it is written, it is making the food vacuole. Okay, this is called food vacuole. A vacuole which is having food called food vacuole, which merge with the lysosome and ultimately digests the food and releases the nutrient for amoeba. Okay, now one more like the example of this one category is macrophages, WBC. They also do such kind of activity. They also do or digest the food in this way only. Okay, now one more term is given over here which is called autophagy. Okay, it means what? Engulf, or you can say eat itself. Okay, it just eat itself. That is called autophagy. Now what does it mean exactly? What happen when there is a cell? Suppose this is the cell and they own some organic matter, it become defective. Suppose it become defective. Okay, now it will create problems for other, and this is the lysosome. Okay, what it will do, it takes the material inside and digest this material and again releases the particles, releases the nutrients which are comes from this digestion. These nutrients again use to synthesize another material. Okay, another material. So it's like a recycling. Like in recycling, what we do, with we just break down the material in a simplest form and again take those simplest form to make another one material, right? This is actually recycling. So here also recycling is going on. Whatever the material is being defective inside the cell, it is taken with the help of lysosome, digested, releases its simplest form, and these simplest form again used to make another one material. So this is how it is used or utilized. So what is doing, it is eating its own material. Lysosome is eating its own cells material. That's why it is called autophagy. Got it, beta?
Yeah.
Okay, so this is about lysosome. Let's have a look how it phagocytosis doing on with this diagram. Otherwise I have told you this one. So what is happening, see, lysosome contains active hydrolytic enzyme, right? Okay, so what it doing, the food, this is the food which is taken inside. Our plasma membrane make us such bulging inside. This is how the food is taken inside. Ultimately a vacuole is formed which is called food vacuole. This is lysosome. It comes to this food vacuole, merge with this food vacuole and ultimately releases its hydrolytic enzyme in this food vacuole, ultimately digests the whole food, and this is how digestion takes place. Got it? Yeah. Now next thing is what, let's talk about the autophagy. So what happened in autophagy? See, here you can see there are two things like vesicle which contain two damaged organelles. There are two damaged organelles are given, mitochondrial fragments as well as peroxisome fragments. Okay, so these two damaged, and now what will happen? Lysosomes come over here. It fuse with the vesicle containing the damaged organelle. Okay, so this is the lysosome. Here you can see peroxisomes and mitochondria. These two organelles are damaged. They are defective. If they are no more required, so they are enclosed in a vesicle. Now this vesicle will fuse with this lysosome. Again lysosome releases its content inside this vesicle and digests both organelles. So it's like a recycling, or you can say cleaning the cell. Okay, it is doing housekeeping work. If there's any defective things, it just clean that defective things. So it is doing the house cleaning. Exactly. Let's go further. Another organelle which is vacuole. See what we are doing right now, we are doing the endomembrane system. I have told you in endomembrane system what are the organelles comes, ER, endoplasmic reticulum, Golgi bodies or Golgi complex, then lysosome, and the last one is vacuole. Okay, so let's do the vacuole. First of all, it derived from the endoplasmic reticulum and Golgi apparatus. Again, it is also derived or it is also secreted from the Golgi bodies. Okay, ultimately the vesicle which are secreted from the Golgi, one of them is vacuole also. So vacuole is like this one membrane and single membrane bound structure, and the membrane which bound this membrane name is tonoplast. Okay, remember this word. So a vacuole membrane is called tonoplast. Its membrane is selective in transporting solutes. Okay, means what? This membrane act as plasma membrane. Okay, it behave as a plasma membrane. So plasma membrane was also a selective permeable membrane, a selective membrane. So this membrane also act as a selective membrane. It doesn't allow everything to go inside or outside the cell. It select the molecules and then only allow it to enter or exit. Okay, let's have a look of their functions. What are the functions? First function is food vacuole by phagocytosis. Okay, so what it do? It help in making the food vacuole. As I told you, this is amoeba, told you just now, and the food is coming inside. So how this food come inside this one? So what happened? This membrane make a this kind of structure. I'll make one more thing. Okay, and this food is taken inside. So this membrane make a covering around this one, and this is how they make a vacuole. It is called food vacuole. So basically it is a vacuole which is having food now, called as a food vacuole, which help in phagocytosis. Okay, another one is called the contractile vacuole. This is also present in the amoeba. So what happen in contractile vacuole? Actually, see, once the food is being digested over here, so what happen? This is amoeba again. So food is digested and all the nutrients are absorbed in the amoeba. The remaining part is called waste. Okay, now this behave like a contractile vacuole. This vacuole again goes towards the plasma membrane. Okay, now this membrane goes towards the plasma membrane and fuse with it like this. Let me show. It fuses with this plasma membrane, and all the waste which are now present inside is being released outside. Okay, so this is how contractile vacuole help in this all these kind of things, right? Contractile means what? Contract. Okay, it just can contract and releases all the things outside. Okay, yes, now let's go for the another word, which is pump excess water. Okay, 1 minute. Yeah, pump excess water out of the cell. It maintains the suitable concentration of ion and molecule. What does it mean? See, as I told you, this membrane act as a selective permeable membrane. So if there's a excess water comes inside this vacuole, what it do? It releases the water outside, so that inside this one, water ion balance, molecule balance, their concentration can be balanced. Okay, so it can do accordingly. If water is higher, it releases the water outside. If there's a ion is very less amount, then it take the ion inside as well. Okay, next thing is what? It do the storage of protein and organic acids. So basically, if I'll say what is the exact function of vacuole, its main function is to storage of water, ions, nutrients, as well as waste material. Okay,
Yeah.
okay, as well as waste material. So these all are things are kept inside. If water is in excess amount in cell, it is stored inside the vacuole. Okay, so now you must be knowing vacuole is, it is a very large in size. In case of plant, it is a very large vacuole. It occupy 90% volume of plant cell. Do you remember I showed you plant cell where a large vacuole is there? If you remember, yeah, can you remember? This is the large size vacuole is over there, which store a large amount of water, ions, so many nutrients are also stored inside this one. Okay, right? So basically its main function is the storage of protein, organic acid. It also contains red and blue pigment as well. Okay, it also has NaCl, I mean like ions also it has. This is called cell sap. When there's a water, there's a ions, okay, then combinedly it is known as cell sap. We used to say cell sap in case of like plant. Okay, this is what I am talking about, plant, not in animal. Okay, because actually vacuoles are actually present in plant. In animal they are very small size, they are present but in a very small size. Now it also responsible for growth of the plant cell, because if the vacuole size is increase, the plant sizes also increase, like this one. They also expand outwards, and this is how growth of plant cell also occurs with, because of the vacuole. So okay, now let's have a look of all the things at once. As I told you, and in endomembrane system 4 organelles are there. What are the things involved in endomembrane system? First of all, there is a nuclear envelope, or you can say nuclear membrane, because from this nuclear membrane only the ER is getting formed. Okay, so it is connected to the rough ER, which is also continuous with the smooth ER. So ER is there, as you can see, ER. From the nuclear envelope the ER is getting formed. Then nearby one more structure, which is the Golgi apparatus. Now the membrane and the protein produced by the ER, it move by the transport vesicle to the Golgi. It entered to the Golgi. Further modification are done over here. It is packaged and then ultimately secreted outside from the transphase. So the Golgi pinch off the transport vesicle and other vesicle that are give rise to lysosome, like lysosome we can have, or it also give rise vacuole, right? So this is the lysosome, this is lysosome, okay, and this also give vacuole, you can say it is also giving rise to vacuole, right? So lysosomes is available for fusion with another vesicle for digestion, it also used for digestion, whereas a transport vesicle which carries the protein to the plasma membrane for secretions. Okay, plasma membrane expand by fusion of vesicle. Proteins are secreted from the cell by exocytosis. Now what do you mean by exocytosis? Means what? I mean like giving out something. Okay, you can just understand it, giving out, it's giving out, right? So this is how the protein is given. I mean like it is not vacuole exactly, don't consider it vacuole. Okay, it's containing the protein which reaches to the plasma membrane, this is plasma membrane, and give all the things like protein, lipid, they are given to the plasma membrane, so the plasma membrane can be synthesized over here. So how it just give out this protein, and this process is called exocytosis. This is called exocytosis, giving something out. Okay, a few question I have given in this one only, just have this have a look of this, and you have to, it's your homework as you can consider. So the 3 questions are there. What role do ribosomes play? So if you now you would be able to answer all these questions. Describe the structure and function, functional distinction between rough and smooth ER, and how ER and Golgi apparatus involved in plasma membrane synthesis. So please do all 3 questions.